Chapter 13 starts our last unit. Our last unit consists of a couple of chapters on molecular biology and then a couple of chapters on evolution. And it may seem strange that evolution follows molecular biology, but it actually makes a lot of sense when you think about DNA as the inherited material in a cell and evolution as a process that works on changing the genetic makeup and the phenotypes in a population. Chapter 13 is very molecular heavy. We go back into the structure of DNA and we talk about the processes related to the replication and modification of DNA in a laboratory, which are very closely linked to the process of replication. So we looked at DNA. We have looked at DNA many times over this semester. We saw it first in Chapter 1 as one of the requirements for living organisms. All living organisms use DNA as their genetic material. This is a universal language that is found throughout all organisms on Earth that are considered living. And the reason that DNA unites them all is that it is the heritable material that was found in the common ancestor that gave rise to all life on Earth. We have also seen DNA throughout our semester. DNA was in Chapter 3. We only talked about it very briefly when we talked about the building blocks of some of our macromolecules, and we talked about the individual pieces that make up a DNA molecule. And then we talked about it again when we got into mitosis and meiosis and the movement of chromosomes and the location of genes on chromosomes. But now we're really going to talk about the structure. And one of the key processes that DNA participates in is the process of DNA replication. Now, DNA is deoxyribonucleic acid. A nucleic acid is a very specific type of molecule, and nucleic acids are really important not only because they code for all of the genetic information in our cell, and not only because they pass that information on to the next generation, but because nucleic acids have a unique ability to reproduce themselves. Encoded within themselves is the template for reproduction. No other molecule can do this. So DNA is really important because it can pass on its own information to the next generation of cells. So let's recap our genetics unit. Remember that offspring resemble but are not identical to their parents. And that some of this has to do with genetic variation that comes out of meiosis and sexual reproduction. But some changes also occur during the process of DNA replication, and they're really important in continuing this concept of genetic variability between generations. Remember that heritable information is passed on to the next generation. So whether you're talking about mitosis and you're talking about the next generation of identical cells, or you're talking about meiosis and you're talking about the next generation of organisms, this heritable information has to be faithfully replicated and faithfully passed on to the next generation. So this idea of genetic replication is critically important in maintaining and passing on genetic information. And then, of course, Morgan showed that genes were located on chromosomes, and Mendel showed that there were alternate versions of genes. We also now know that the inheritance of those genes and those characteristics follow the movement of chromosomes. So by the early 1900s, most biologists accepted that chromosomes were the heritable material within a cell, and that it was that traits were encoded within those chromosomes. A lot of what we understand in DNA replication comes from studying bacteria, specifically E. coli. E. coli is probably a bacteria that you are familiar with. It often gets a bad rap because it causes food poisoning, but in fact it's just a bacteria that's found in our gut. All healthy guts have them, and they are not doing any harm. In fact, they're doing you a lot of good, helping you digest your food and things like that. But because they're readily available, E. coli has been extensively studied with respect to molecular processes within a cell. So DNA replication was originally looked at in E. coli. E. coli chromosomes are a little bit different than ours. E. coli chromosomes are a single small loop of DNA. When I say small, I don't mean tiny, I just mean small compared to human chromosomes. But they are circular instead of linear. Human DNA, remember, and most eukaryotic DNA are long linear pieces of chromosomes. Whether we're talking about E. coli or human cells, a lot of genetic information must be packed really tightly into the nucleus. In fact, the nuclear content of a single human cell is about 4 centimeters long, which doesn't sound like very much, but considering that that has to be condensed into the nucleus of a cell that you cannot even see, that's quite the feat. So both E. coli chromosomes and human chromosomes, and all chromosomes really, are packed in a very specific way. This diagram on the bottom shows some of the packing that goes on. You have your DNA double helix. It's wrapped around proteins, and those proteins are wrapped very tightly and condensed into loops. I think we talked about this when we were talking about chromosomes before. If you think about holding one shoelace, one end of a shoelace in one finger and in the other hand twisting it up really tightly, and it sort of coils up back on itself, that's how our chromosomes are packed into the cells. So it was established early on that chromosomes were the vehicles, basically, that were passing genes on to the next generation. But there was a lot of question about whether or not DNA or proteins were the genetic material that contained genes. In 1928, a man named Frederick Griffith, he was working in the UK, was working with viruses and mice, and he found that if he mixed pneumonia viruses that had been killed and transferred them or exposed them to live cells, some of those cells would actually pick up sort of a component that could cause pneumonia in mice. So even though the bacteria that causes pneumonia was actually killed, there was something that was being passed on that could cause mice to contract pneumonia. This diagram shows Griffith's experiment. These mice that he worked with were injected with the bacterium that causes pneumonia. So understandably, if you are injected with pneumonia bacteria, you're going to get pneumonia, and these mice who were exposed to those bacteria died. So we see that on the left-hand side there, and that's exactly what you would expect to happen. As a control, healthy mice were injected with bacteria that do not cause pneumonia. Then they were injected with bacteria that had been heat killed. So these bacteria were no longer able to pass pneumonia on to the mice, and of course those mice survived and were healthy. But then he decided to mix the cells together. And he found that if he mixed the heat-killed cells with the living cells, the mouse would die. And from those mouse, he found living S cells, so the cells that originally killed the mice that contained the pneumonia bacteria. So this told him that there was some sort of factor that could be passed from those dead cells to the living cells. Continuing on, about 50 years later, a man named Erwin Chargaff was looking at DNA from several species, and he found something really interesting about it. He found that DNA composition varied by species. So, for example, the sea urchin might have 32% adenine, which is one of our nitrogenous bases, and E. coli might have 24%. So there was some variation in the composition of DNA. But he found something very interesting. He found that no matter what species, the amount of adenine in the DNA molecule always equaled the amount of thymine in the DNA molecule. He also noticed that in a particular DNA molecule, the amount of cytosine always equaled the amount of guanine. So at this time, a lot of people were looking at DNA and proteins, and they were trying to figure out what Frederick Griffith's transforming factor was. Were chromosomes passing on genetic information in their proteins or their DNA? So to finally figure out which component of chromosomes, the proteins or the DNA, were actually passed on as heritable genetic information. We have an experiment in 1952, which is about the time that Watson and Crick were looking at the structure of DNA. It's about the time that Erwin Chargaff was looking at the structure of DNA. So Hershey and Chase were a married couple who were working together, and they were studying the T2 phage. Phage is short for bacteriophage, which is a virus that infects a bacteria. So if I say phage, I mean virus. This T2 phase was specifically infecting E. coli. So again, we're using E. coli as our model organism here. And they wanted to know if that bacteriophage was passing on DNA or proteins as the infectious genetic material. So I'm going to take just a minute to explain briefly the structure of a virus. Down here we have an E. coli cell, that's a bacterial cell. Up here we have this funny, almost spaceship or alien looking structure, that is the bacteriophage. Up here in the head of the phage we have DNA. Now that is not a nucleus, that is just DNA packaged into a protein outer layer. So all of this green is a protein. DNA is in the head. And this virus works by clamping on to the cell with these little spindly legs here and injecting DNA basically like a little molecular syringe. Or, well, actually, at the time they didn't know it was DNA, but we now know that it's DNA. So the bacteria recognizes the virus recognizes a particular bacterial cell and injects the genetic material into it. That causes the cell to then produce new viruses, and that cell bursts. The viruses are released and they go on to identify another host cell, inject their DNA, and the process happens over again. So Hershey and Chase wanted to determine if the protein from that virus, the outer coat, that outer protein coat, or if DNA was the genetic material that was being passed on. Which of those was the material that was causing the infection and causing the production of new viruses? So they did two parallel experiments. In the first cell, they used radioactively labeled proteins. They used sulfur, radioactive sulfur label, in their proteins, and they essentially labeled all of the proteins in that virus. They allowed the virus to attach to the E. coli cell, to inject its DNA, and then they sort of agitated it just using a blender. And by doing that, they were able to knock the viruses off the cell, but not burst the cells. So then they separated the cells from the liquid with the virus in it and found that all of the protein, the labeled radioactive protein, was in the solution around the cell. So they found that the protein was not in the cell. In a second experiment, they labeled the phosphorus in DNA with a radioactive label. And again, they let the virus inject its DNA into the cell, or inject DNA, and agitated it, knocked the viruses off the outside of the cell, and then looked to see what was left in the cell, or where their radioactively labeled information was. They found that the pellet in the bottom, when they spun it really fast and separated everything out, they found that the radioactive DNA was still in the portion where they expected the cells to be. This was really important because it told them that DNA was the genetic material. DNA was the molecule that those proteins were injecting into the cell that was causing the viral infection. So this is our first evidence that DNA is actually the genetic information molecule. Now keep in mind, up to this time we knew about chromosomes. We knew that somehow our genetic material was encoded on those chromosomes. We didn't know if that genetic material was the DNA or the protein.